Dr. Apkarian is Professor of Physiology, Anesthesiology, and Physical Medicine and Rehabilitation at Northwestern University in the Feinberg School of Medicine. As well as doing groundbreaking work on consciousness, Dr. Apkarian has developed a field, or been instrumental in developing a field that we've already heard about in the excellent presentations earlier, relating pain to learning and expectation. And he put forward the idea that chronic pain is actually a result of emotional learning, which links it to the field of reinforcement learning, which I think is a really important insight. Today, his talk is Brain Mechanisms of Chronic Pain, Translational Opportunities. Thank you, Dr. Apkarian.
Thank you, Paul. Okay. So thank you. It was fun listening to all the speakers before me. I will try to stay on time and give you an overview of some of the work we've been doing for more than 20 years. I would like to emphasize the translational approach of our research, and in that sense, I will, throughout the talk, I'm going to switch back and forth between human studies and animal studies. And overall, I don't want to go through some of these introduction. Lauren already introduced the topic of acute and chronic pain, and all the other speakers before me. Obviously, it's a huge problem worldwide, and it's in urgent need for novel and more efficacious treatments. We don't have acceptable treatment options for chronic pain patients in general. And our general overall approach is that this is fundamentally due of our lack of knowledge about specific mechanistic proper processes that underlie chronic pain. And I start with René Descartes' drawing of pain perception in a sense that within this transduction, transmission, and encoding, there's now extensive data that as soon as pain becomes a persistent state, there's reorganization at the end organ level, at the receptor level, transduction level. There's reorganization and increased sensitization of transmission within the spinal cord. And we have now shown more than 15 years ago that there's, in fact, both brain anatomical and functional changes in patients with chronic pain. So the general question has been that we've been pursuing or chasing for many years is the chicken and the egg. Who is more responsible to what? How much of this plasticity controls the pain perception, and what are the critical parameters within that system? And I'm going to give you some overview of some of the general concepts that we have advanced, and pick and choose some new ideas that I am at least excited about most recently. So overall, I want to say that we can now, this is my general outline, that we can generate both diagnostic and prognostic biomarkers of chronic pain. And that I want to emphasize the point that we can go back and forth between human studies and animal studies and directly interrogate physiological properties of circuits that we identify in the clinical condition, and then look at the mechanistic evolution of events within them. Today, I want to emphasize specifically just two brain regions, primarily nucleus accumbens and the hippocampus. And I want to show you some of the data that both of them are causally involved in controlling chronic pain. And the evidence is, in fact, complementary both from humans and animal studies as well. And I want to show you just a little bit about how these things, in fact, do translate into potential novel treatments as well. So very simply, this is our overall concept, which is a very simplistic idea in that there is 4 different stages for chronic pain. The chronic pain has, as similar to all chronic conditions in general, chronic pain would have risk factors, predispositions. Then these risk factors, together with an injury event or a stress, a combination of stress and or injury, will interact with each other to turn into a transitional state. And this transitional state in time would either result in the pain going away, healing process, or if the pain is maintained, it develops into a chronic pain state. Just terminology-wise, we think of this initial injury as the acute event, this transitional state as subacute pain condition, and in time, this will turn into chronic. We have spent a lot of time studying this mostly in chronic back pain patients, but complementary similar results are observable in other conditions as well. So the classic viewpoint is that all of these stages are explained by nociceptive circuitry, in the sense that the sensitivity of primary afferents and spinal cord reorganization, spinal cord sensitization is adequate to explain pain. Our position, which we've developed over the last 20 years, I just want to give you an overview, is that the limbic emotional circuitry define risk, that the properties of this circuitry define the predisposition, and learning processes, emotional learning processes define this transitional state, and that the maintenance state is in fact a new, that the chronic pain is a new brain state with distinct anatomical and functional properties. I will show you some of the data along those lines throughout my talk. With this general concept, we can then actually define more specific biomarkers. In a sense, any predisposition properties can be thought of as prognostic, while the maintenance properties, the state of markers that are related to the final state of being in chronic pain, would be diagnostic. And we think of this transitional state as a window of plasticity within which preventions may be optimal, and I will show you some data along those lines. Just to summarize our work over the last some 20 years, we see from the predisposition part, we see brain white matter properties, brain limbic structural properties, and genetic properties. All of these things come up as risk factors. And we think of this transitional state as the sum of 1 plus 2, the injury plus the predisposition. And this, we think of this as a mesolimbic learning process. And this learning properties defined fundamentally by the interaction between medial prefrontal cortex and nucleus accumbens as defining whether the subject is transitioning into chronic pain or transitioning away from chronic pain into healing. In the maintenance state, I will not show you much data here. The brain activity for pain is distinct from that of acute pain. Brain anatomical properties are different, and information processing in the brain is globally disrupted. The brain, we can actually calculate the impact of disruption of global information processing in these patients. Let me show you the same concept from a circuit diagram point of view. This is the limbic brain in humans. There's a beautiful corresponding circuitry in rodents. As a result, we can go back and forth between them. But in the normal everyday life, nociceptive signals invade both the limbic as well as the cortical regions. And we think of this as, in fact, nociceptive signaling as protecting tissue from injury. And in fact, when there is an injury that's above and beyond that protection, there is a failure of protection, and that we define as conscious perception of pain. And within this, when this injury state is persistent, there are two options. Either cortical circuitry can suppress the emotional response to that injury, and then the subject heals and pain goes away. Alternatively, if this, in fact, instead, the cortical limbic circuitry enhances the learning process, then the system, at least through dopaminergic learning processes, will then feed back to the cortex and reorganize the cortex into a new emotionally reorganized new state of chronic pain. So I'm going to give you just a single example of the predisposition model. In a sense, if it's a predisposition, it's a prognostic biomarker. It should be pre-existing. It's a pre-existing risk. It should predict transition. since it's predicting the future, we have to call it as a causal event. We would like it to be validated, and since it is a causal mechanistic process, it should reveal mechanistic events in general. And importantly, since this is a predictor, it should be invariant of changes in time. Let me show you my favorite example. This is from, again, a study we have published many years ago of looking at subacute back pain patients, looking at their brains over many months, over multiple years, in fact, and looking for predisposition. This would be basically properties of the brain at time zero that would that can predict who 1 year later will be either persisting or recovering. So everybody comes in undifferentiated. In time, they differentiate into two groups, and that differentiation we should be able to detect at time zero. This is the pain properties of these patients. The subacute back pain patients, they are by design enter the study at the same level of pain. Over 1 year, half of them continue to decrease their pain. These are the recovering people, while the other half are persisting, and so these are becoming chronic pain patients. So this differentiation between pain, if we can predict at time zero, those are risk factors. And our primary risk factor is, in fact, the nucleus accumbens medial prefrontal cortex interaction. Basically, this is a single number that we derive from each subject, the amount of correlation between these two areas of the brain. At time zero, the subjects who are persisting have more interaction, while the ones who are recovering, and that difference is constant, does not change over time, over a whole year, even though the pain is changing. Okay, and in fact, that value at time zero can predict robustly who is moving into becoming a chronic pain patient, both at 3 months, 6 months, and 1 year, at about an 80% accuracy, with an independent validation approach as well. And just to sort of complete that story, I will not go through the details. We can, if we examine the whole limbic network, both from anatomical properties and functional properties, we can identify independent pathways, all of them contributing to this model, which predicts with about 60% of the variance of who will become chronic pain patients. Let me move on to some animal data. Since this accumbens is a region that is involved in addictive behavior in general, it is a region that we are very much interested in. And Lauren briefly mentioned these animal models that we have for studying chronic pain. Our favorite model is this spared nerve injury. This is simply a partial injury to the sciatic nerve, and these animals show behavioral signs of being in pain for the rest of their lives. So we use these animals to study what's happening within this transitional state, in a sense that the design of these animal models cannot give us clues about predisposition, because every one of them is developing chronic pain. But we can look in time what's happening within that time window. I want to show you some data from nucleus accumbens itself. These are patch clamp recordings in TD tomato animals, where the red and the green are D1 and D2 receptor containing spiny neurons within the shell region of nucleus accumbens. This is the emotional portion of nucleus accumbens. And we can see that in animals, neuropathic injured animals, excitability of the D2 receptor neurons is increased compared to sham animals. The D1 receptor neurons don't change at all. The dendritic field of these neurons shrink within 5 days after the injury. The synaptic inputs to the region are decreased in the neuropathic animals compared to the sham animals. And in fact, the dopamine levels, the actual concentration of dopamine in the region is decreased by 50%, And the main input to this region is the ventral tegmental area from the brainstem, and in fact, its response properties, and in fact, it's the source of dopaminergic input to the region, and it's reflecting the decrease in dopamine itself. Excuse me. So we can manipulate these neurons by chemogenetic manipulation, and we can either increase their excitability or decrease their excitability. And by simply changing these excitabilities, we can change neuropathic pain behavior. So the simple level of excitability of these neurons control neuropathic pain behavior. Therefore, these neurons are causally linked to neuropathic pain. Given these results, we recently did a small clinical trial, and this is basically, I'm going to show you a few slides on this. And the idea is to bring in subjects in this transitional state, the subacute back pain patients, treat them for a long period of time by simply giving them back dopamine and an NSAID to replenish their dopamine, and see if that would in fact block the transition from subacute to chronic pain. And since the nucleus accumbens and in fact the limbic brain in general has gender dependence, we also thought that there should be a gender dependence in the outcome. This is the overall design of the study. I will not go through the details of the study, but basically you have 59 subjects who finished the study, and the subjects either were treated by dopamine plus naproxen or placebo plus naproxen, or no treatment at all. The primary endpoint of the study, in fact, was not validated, in that we did not see a difference between the dopamine plus naproxen versus placebo plus naproxen. However, we do see a sex-dependent, beautiful difference in the pain relief, and it's shown here. Here, the gray bar is where the subjects are being treated, either with L-DOPA plus naproxen or placebo plus naproxen, and the pink line are the females, while the blue are the males. And there's a very nice interaction between type of treatment and the effect of pain relief. And you can see that in the females who are being treated with the L-DOPA plus naproxen, pain relief is about 80%. Okay, it's a huge number. And after stopping the treatment, these effects persist for another 3 months. And in fact, we can show, again, if you simply look at this accumbens MPFC connectivity at entry into the study, the females show lower connectivity than males, and 6 months later, there is a complete interaction between treatment and gender. So in a sense, we can see correlates of the treatment in the single brain region with this treatment. So the data is small, so we are hesitant about the overall effects, but it is validated with brain biomarkers, and it needs to be replicated. I'm going to briefly switch to the hippocampus, and the general idea is chronic pain as accumulation of a memory trace that's not being extinguished. This was our general initial hypothesis, and now we have generated a lot of data. I'm going to show you some of the results both in humans and animals. From the human studies...
Sorry, Dr. Apkarian, just to update you, it's just a couple of minutes to the end of the session, so...
I will be done.
Great, thank you.
Yeah, so in
Sorry, I hate
studies,
doing this.
That's fine. We can see that connectivity of the hippocampus is different between different subacute and chronic back pain patients. And in fact, that connectivity reflects the changes in pain over a 1 year time period. And now we can also see that in fact there is anatomical changes in the hippocampus. The anterior portion of the hippocampus, in fact, is distinctly changed in shape, and that's specific to females. And this thing is a time-dependent event in that it develops over multiple years. And let me skip this slide for the sake of time, and simply show you that, in fact, we can also see in animal models that neurogenesis is both affected by neuropathic pain injury And blocking neurogenesis, in fact, delays the development of neuropathic pain itself. And in fact, if we actually stimulate the dorsal hippocampus, we can reverse neuropathic pain behavior in multiple animal models. And we can also change their motivational responses as well. So again, the hippocampus is causally linked, both with activity and with neurogenesis. Primarily, the dorsal hippocampus relieves pain-like behaviors. And just to go back to my starting slide, in a sense that we can think about these components from different types of chronic pain, that the relative contribution of the injury versus the risk may be differentially thought of as distinct processes with which we can think of different chronic pain conditions. And I will stop there and actually thank all the people, all the members of the lab, as well as the NIH funding that we have had for many years. Thank you.
Thank you. I feel isolated clapping on my own, but as I say, I'm sure everybody, I'm clapping on behalf of nearly 1,300 people. Thank you so much for such an elegant talk. We have about 20 seconds left to the end of the session, so I'll just pass on one question actually from Dr. Mancini. Regarding the beautiful nucleus accumbens rodent study, is there any evidence of modulation of value-based learning in relation to the key result of pain modulation?
It's a great question. We haven't asked the question specifically. On the other hand, I mean, we do have human studies where in fact, acute painful stimuli show very different, massive differences in their responses within accumbens, which we think would be consistent with that idea. Yeah, of course.
Thank you. Okay, well, it just leaves me, because we have to draw to a close, I'm sorry to have cut people off. I would have loved to have heard a lot more. Thank you so much to our really wonderful speakers. Thanks for an excellent session, and I hope people have enjoyed it. Thank you.
Thanks for organizing it, Paul.
My pleasure. A real pleasure.